Hello, this is Matt Dean with A Plus College Ready, and today we're going to talk about the box and whisker plot. The box and whisker plot is a new kind of statistics-related graph that's been added to the AP Biology curriculum. So a box and whisker plot, or sometimes it's also called a box plot, is a way of summarizing a data set measured on like an interval scale. It's often used to explain the data. It's used in explanatory data analysis. It's used to show the shape of a data distribution. It's also good at looking at the central value, like the median of the distribution, and it also shows the variability of the distribution. And you can see a box and whisker plot down on the bottom of the screen. They can be drawn horizontally, like the one shown here, or they can also be drawn vertically. So to create a box and whisker plot, you first of all order or rank the raw data in numerical order, starting at the least, going up to the greatest. You then find the median of the data set. Now remember, the median is the middle number. That's assuming you have an odd number of data points. If you have an even number of data points, you take the two middle numbers and you average those to get the median. After you find the median, you divide the data into two equal halves at the median. Remember, the median is the middle value. If the median is a data point, you don't include that in either group. Next, you find the median of the lower half, just the lower half of the data. The median of that lower half is called the first quartile, or Q1. It's the equivalent of the 25th percentile of the data. Next, find the median of the upper half of the data set. This value is called the third or the upper quartile. We'll usually call it Q3. This is equivalent to the 75th percentile of the data. Step 6, on an appropriately scaled number line, draw short vertical lines at the values for the lower and the upper quartiles. Next, you're going to complete a box that joins these two vertical lines. This box represents the middle 50% of the data. You're going to mark the median as a vertical line inside the box. Finally, you're going to draw the whiskers. The whiskers are the extremes. So you're going to go out to the lower extreme value and the upper extreme value. You're going to draw dots or lines, and then you're going to extend a line from each side of the box to each of those extreme values. So let's look at an example. Actually, before we do, let's talk about some special issues related to box and whisker plots. Oftentimes, you'll put two box plots or box and whisker plots on the same graph. We call those parallel box plots or parallel box and whisker plots. They're a good way of comparing different groups of data. Let's talk about another issue related to the box and whisker plots, and that's called the interquartile range. This is the difference between the upper and lower quartiles. So you take Q3 and subtract from it Q1 to get the interquartile range. This essentially gives you the spread of the middle half, the middle 50% of the data, and it's a good way to evaluate variability. A higher interquartile range means there's more variability in the data. Sort of a rule of thumb is that 0.75 times the interquartile range is a good way to approximate the standard deviation of the data set. So a couple more special issues related to the box and whisker plots. So when you're comparing two different box and whisker plots, one thing to look at is compare the locations of the interquartiles. If the interquartiles, which are the two boxes that you're comparing, if they overlap by a full quartile or more, this indicates that there's probably not a significant statistical difference in the two data sets. If the two interquartiles, the boxes, don't overlap at all, or if they don't overlap by at least a full quartile, there probably is a significant statistical difference between the two data sets. Another thing we want to talk about is where's the median line relative to the first and third quartiles. This is a good way to measure the skewness or the asymmetry of the data. If the distribution is symmetric, the median is going to be exactly between Q3 and Q1. If the median is closer to Q3 than to Q1, the distribution is said to be negatively skewed or skewed to the left. This means that the left tail of the distribution is longer. And this tells you that the data points on the right side of the median have less variability than those on the left. If the median is closer to Q1 than it is to Q3, the distribution is said to be positively skewed or skewed to the right. And this tells you that the data points on the right side, those have more variability than the ones on the left side. So here we see a left skewed graph up at the top, or negatively skewed. Here we see a symmetric set of data. And finally, we see a right skewed or positively skewed data set on the bottom. So one more set of special issues. Let's talk about outliers. So an outlier is an extreme value that's not typical of the data. These are values that lie more than 1.5 times the interquartile range away from the nearer quartile. Outliers are usually represented by open circles or asterisks, and the whiskers are then drawn to the next closer value that's less than 1.5 times the interquartile range. I don't think we'll run into this issue very much in AP biology. I don't think we're going to get a lot of outliers. One note of caution, though, it is important that you as students understand that a smaller side of the box or a shorter whisker, that doesn't mean that there are fewer values represented in that part of the box. It just means that the values are grouped closer together. In other words, the smaller side or the smaller whisker has a smaller spread than the larger side or the larger whisker. So here's an example. A researcher counted the number of trichomes. These are hair-like structures on Wisconsin fast plants. They used 15 plants, so that's the sample size. Here are the number of trichomes. So we want to construct a box and whisker plot for this data. So as we said before, first of all, we're going to order the data from smallest to largest. And you can see I've already done that for you here. Next, we're going to locate the median of the data. Well, here we have an odd number of data points with 15. So we're going to go to the 8th data point in the middle. Our median is 20. Next, we're going to divide the data into lower and upper halves. And you can see that I've color-coded that here. So we've got the blue half and the green half. And remember, if the median is a data point, you don't include it in either half. So 20 doesn't get in that 20, the red 20 doesn't get included in either half. Next, I'm going to find the median of both the upper and the lower halves of the data set. Both of those have odd numbers of entries. So the median of the bottom set is 15. The median of the top set is 22. The median of that bottom set is the first quartile, or Q1. And the median of that top set is the upper quartile, or Q3. So if we want to find the interquartile range, you can see how we do that here. The interquartile range is the Q3 value, 22, minus the Q1 value of 15. So we end up with an interquartile range of 7. So for an outlier to exist, it has to be 1.5 times 7. So it has to be more than 10.5 from the box, from either edge of the box. So we'll keep that in mind when we're drawing our box. So next we're going to construct our box and whisker plot. So let's go to a graph and do that. So first of all, we're going to draw vertical lines that represent Q1 and Q3. So Q1 is 15. So there's Q1. Q3 was 22. So we're going to now connect those up and make a box. We're going to draw in the median. Our median was at 20. So there's our median, like so. Next, we want to put in our outliers. So our highest value was 26. That's our upper extreme value. So we'll draw that in like so. And our lower extreme value was at 10. We'll draw that in like so. Let's check and make sure those aren't above the, those aren't outliers. So remember, it's got to be within 1.5 times the interquartile range. So that was 10.5. So this is way less than 10.5, the difference between this side of the box, 22, and that extreme value of 26. Here, this is 15 on the left side. This is 10. So also that's way less than 1.5 times the IQR. So to finish up our box, we draw in horizontal lines that connect our extreme values to our box, like so. And we've got some straight lines, so let's see if I can clear those up just a bit. And we also want to mark in our median. I think I already did that, but I erased it. So there's our median right there. So this is our median. This is Q3. This is Q1. This is our lower extreme value. And this is our upper extreme value. And notice we could say because the median is closer to Q3 than it is to Q1, that this particular data set is skewed to the left or it's negatively skewed. All right, we're going to stop there and we'll look at another example problem here in just a second. Here's an example of that same box and whisker plot drawn vertically instead of horizontally. Again, you can see the lower extreme point. You can see Q1, the first quartile, the median marked right here in the middle. Here's Q3, and here's the upper extreme at 26. So again, you'll see those very oftentimes drawn horizontally, but they can just as easily be drawn vertically. So let's look at another example. Here's sample 2. So the researcher then artificially selected the 5 plants with the most trichomes from the first group of data that we looked at a second ago, and allowed only those plants to reproduce with each other. After a period of growth and development, the researcher counted the number of trichomes on the generation 2 offspring and found the following number of trichomes on the 15 generation 2 offspring. So here's our data for generation 2. So we want to construct a box and whisker plot that depicts both the generation 1 and generation 2 data on the same graph. That way we can compare them. So just like before, the first thing we're going to do is order the data from smallest to largest. And you can see that here from 21 up to 46. Find the median, the middle number of the whole data set. That median is 38. Then we're going to divide the data into upper and lower halves. Here's the lower half in light blue, the upper half in green. And notice since 38, the median is an actual data point, it's not included in either set. So now we're going to find the median of both the lower and the upper halves. So for the lower half, that median is 24. That's now Q1, the first quartile. And the upper median is 42. And that's Q2, or Q3 rather, the third quartile. The interquartile range is the difference between Q3 and Q1. So Q3 is 42. Q1 is 24. We subtract those and get an interquartile range of 18. So now we want to construct that box and whisker plot. So I'm going to go back to my original graph. And you'll notice that I moved that graph to a bigger scaled piece of graph paper. So here's my generation 1 graph. And now we're going to graph generation 2. So we'll start off by plotting the first and third quartiles as vertical lines. So I'll do that here. So my first quartile was 21. So 21 is going to be somewhere right in here. My third quartile was 42. So that's way over here, 42. I want to connect those and make a box. So connect those with horizontal lines. There's my box. I'm going to draw in my median at 38. So we're really skewed left here. I'm going to draw in my upper extreme at 46. Here's my upper extreme. My lower extreme's at 21. I did something wrong here, guys. Let's start back over with that box. So the Q2 box should have started at the Q1, which is 24. So that's Q1, 24. Q3, 42. Connect those up like so. Drawing my median at 38. Drawing the upper extreme at 46. So here's that upper extreme. I'll go ahead and draw in the whisker there as well. And my lower extreme was at 21. 21 is going to be somewhere right in here. And I'll draw in that whisker. All right, so there's our two data sets. Again, this is Q3 right here. This is the median. This is Q1. This is the lower extreme. And this is the upper extreme. All right, so now we have parallel box and whisker plots. Box and whisker plots are box plots on the same axis. So if we go a little further, we've got some questions here to answer. Are the two data sets significantly different, and justify your answer? So the way you want to look at that is do the boxes overlap? And notice that this box does not overlap at all with this box. So if the boxes don't overlap at all, we can say there's a good chance that these two sets of data are statistically significantly different from each other. They're significant in a statistically significant way. They're different in a statistically significant way. Now, if they did overlap some, we said that they need to overlap by somewhere around a full quartile or more. And if they do, we can say they're probably not statistically significant. The difference is probably not statistically different. All right, so that's box and whisker plots. I hope that helps.